features of gene signatures, we'll discuss some issues related to gene signatures construction, and we'll briefly discuss feature selection, which is one of the main steps of constructing a gene signature. So gene signatures are similar to individual biomarkers, but as they comprise of several genes, gene signatures are generally more informative and, what is important, they are more stable to experimental, for example, lab-level inaccuracies. So inaccurate measurement of, for example, gene expression level is more crucial in case of individual biomarker, but can be compensated by more accurate measurements of gene expression levels in case of gene signatures that comprise of several genes. Similar to biomarkers, gene signatures have standardly may have three aims. There are diagnostic, prognostic, and predictive gene signatures. Diagnostic gene signatures are used to test whether a patient has a certain disease. Prognostic gene signatures allow forecasting a risk of getting a disease, allows predicting risk of recurrence, allow to estimate recurrence-free survival, and so on. Predictive gene signatures allow to predict whether a given drug or a given therapy scheme would be efficient for an individual patient. So, in all the cases, input data for such prediction are omics features of genes, for example, expression levels of genes, mutations, different epigenetic modifications can be also used as input data for gene signatures. Yet, standardly, in the majority of cases, gene signatures utilize expression levels of genes or SNPs, so single nucleotide polymorphisms. Two standard examples are genes breast cancer associated gene 1 and 2, BRCA1 and 2 genes. Mutations in these genes are related, at least statistically related, to essentially high risk of breast cancer disease, of ovarian cancer disease, and identification of mutations in these genes may even lead to a surgery before identification of cancer, for example, of breast cancer, so just preventive surgery. While other well-known gene signatures associated with breast cancer are Oncotype DX and MAMAprint. These are transcriptomic gene signatures, so they are based on expression levels of genes, and they predict the risk of recurrence-free survival for at least 5 or 10 years for patients with estrogen-positive breast cancer. It is worth noting that precision characteristics of gene signatures are generally very far from 100%. So gene signatures are not standardly used as the only base for making a decision. They are standardly used just as an additional source of information that is added to multiple other sources and interpreted by a doctor. A good example here is related to precision characteristics of, for example, breast cancer recurrence risk signatures, where for a low risk group, a risk of recurrence is standardly between 5 and 10%, while for a high risk group, this risk of recurrence is standardly about 50%, or in some cases even 30%. So high risk of recurrence does not mean that the probability of recurrence is almost 100%. It can be much lower. So just to make this example even more obvious, here we can see curves related to oncotype DX gene signature for prediction risk in colon cancer, not in breast cancer, but in colorectal cancer. So this gene signature stratifies all patients into three groups: patients with high, intermediate, and low risk of recurrence. And as one can see here, the risk of recurrence in a low-risk group, the true risk of recurrence, is 12%, while this risk of recurrence for the high-risk group is only 22%. It means that statistically, groups identified by the gene signature are really different, but in fact the difference is not really huge, and these risks are relatively close even in high and low risk groups. So clearly it's better to have a gene signature with high precision, but in many cases it's a real problem, a real unsolved problem in many cases, of constructing gene signatures with high precision characteristics. Regarding precision characteristics, the main ones are sensitivity, specificity, positive and negative predicted values. Sorry, a misprint here. Positive and negative predictive values. And one more misprint here. So generally, all patients in a test, in a validation experiment, all patients can be divided into four groups. True negatives, patients that have negative answer of a test system, that belong to a negative class, whatever this negative class means, that are predicted as negative by our gene signature. So these patients belong to a group true negatives. Similarly, patients from a positive class, from a true positive class, predicted as being in this class are true positives, but there are two types of errors. False positives, so patients from a negative class that are classified as positive by our classifier, and patients that belong to a positive class but are classified as negative. So we also have false positives and false negatives. And the sensitivity, the value of sensitivity, is number of true positives divided by the sum true positives plus false negatives. This sum true positives plus false negatives is the total number of patients in the positive class. So in fact it is the fraction of patients in the positive class that are also classified as positives. Similar specificity is the number of patients classified as negatives divided by the total number of patients in a negative class. So is the fraction of patients in a true negative class classified as negative. These values are important for, a population level analysis, but they are not so informative for individual patients. For an individual patient, more informative characteristics are positive and negative predictive values. What are these parameters? Positive predictive value is the number of true positives divided by the sum of true positives plus false positives. So it is the fraction of true positives in the complete set of patients classified as positives. And similarly, negative predicted value is the number of true negatives divided by the total number of patients classified as negatives. So what it means for an individual patient? It means that if a sample related to this patient was classified, for example, as positive, then the probability that this classification is correct can be estimated by these positive predictive values. Similarly, if a sample for a patient was classified as negative predictive value gives a probability that this classification is really correct. Clearly, these simple characteristics can be used in case of binary classification. In case of two-class classification, if we have more classes, a bit more complicated characteristics of gene signatures, class-related or related to the whole gene signature, should be used. So standard usage of characteristics like sensitivity, specificity, positive and negative predictive values, in most cases, is associated with binary classification. Regarding the classification algorithm itself, again, in the majority of cases, all samples are treated as points in multidimensional space. For example, if we're working with a gene signature that is based on expression levels of genes, that is, we're working with transcriptomic gene signatures. Then for each gene we have an expression level, and the dimension of space we're working with is equal to the number of genes in our, well, to the number of main genes in our gene signature. And the coordinates of a point are simply expression levels of genes. And clearly, multiple supervised machine learning approaches can be used to construct a classifier. So we'll just go over the simplest approaches here. One of them is an approach called k-nearest neighbors. In many cases, r equals even 1. So we have our learning set, that is, we have patients with known answers. We know for these patients to which class they belong to, and we have one new patient for which we should perform a classification, for which we should give an answer, and we should attribute this patient to one of the classes. So what can we do in case when we're treating all patients, in case when we're treating all samples as points in a multidimensional space? So we can simply find the closest samples in our learning set and then perform voting. So for example, if k equals 3, we can find 3 nearest neighbors. Here we can see that the majority of these neighbors belong to the yellow class, and thus k nearest neighbors classified with k equal 3 would attribute this patient to a yellow class. Note that mathematically there are multiple ways to define a distance function. We can use standard Euclidean distance, we can use Manhattan distance, there are multiple other distances. So in fact, it's not the approach specified in all the details. It is, k-nearest neighbors. It is just a scheme where one can vary such parameters as k, where one can vary the approach to measuring distances, and varying these parameters, different classifiers can be obtained that give, in some cases, different answers. So optimization here is required. Another very simple method for constructing a classifier is a nearest centroid method. It performs the following. For each class, we compute a centroid, so a center of this class, for example, as a coordinate-wise mean for each class. So for each class, we, well, in some sense, forget about individual samples, but we have the standard, the central point describing this class, centroid, for example, center of mass. And then, when we have a sample, we simply find the nearest centroid, again, nearest from the point of view of a selected distance function that's not always a Euclidean distance function. And we attribute a sample to a class represented by the nearest centroid. So again, yellow in this case. Well, clearly, in some cases, k-nearest neighbor classification and nearest centroid classification give different answers. For example, here, if k equals 1, the nearest neighbor is a yellow point, but a nearest centroid the nearest centroid is green. So varying a scheme for constructing a classifier clearly affects the result of a classification. Another method that's a bit more complicated than k-nearest neighbors and nearest centroid is known as support vector machine. So what it does in the simplest case, when there are two classes and classes are linearly separatable, well, and in case of linear kernel. So in case when classes can be separated by a hyperplane, for example, by a line in a 2D visualization, really there are multiple lines that can separate these classes. And the question is, what line, what separation is optimal? A logical approach, very clear approach, is to try to find a line that makes the gap between classes as wide as possible. And it turns out that this line can be found in a relatively simple algorithmic way. In case when linear separability is violated, we can still try to find a separating hyperplane which aims at making the gap as wide as possible, but which penalizes points or samples from a learning set that belong to an invalid zone, to a gap zone, or even to an invalid zone. So support vector machine in this case try to construct wide separating zone, still having as low number of errors as possible, and there's a coefficient that tries to find a balance between the width of a separating zone and the number of errors, as clearly we can have very wide quasi-separating zone but with very many errors. So there's a standard coefficient inside support vector machine that balances the width and errors. So if we try to compare support vector machine approach with nearest centroid approach, we see that nearest centroid also separates two classes by hyperplane defined simply by centroids. So hyperplane orthogonal to a segment that goes from one centroid to another and orthogonal to this segment. And support vector machine constructs, well, in a way, an optimal separating hyperplane. So it seems that support vector machine is a better way for constructing a classifier, yet it holds in cases when the size of the learning set is sufficient. In case of insufficient sizes of learning set, simpler methods may perform better than more complicated methods. Well, clearly, linear utilization of support vector machine is only a simplest one, as there are nonlinear kernels in this type of classification, and these nonlinear kernels in fact bring points from a space with lower dimension to a space with higher dimension. In the example in this slide, well, red points are not linearly separable from green points, yet if we introduce one more coordinate, well, for example, if we select an additional point somewhere here, so an additional point near the centroid of red class, and add a third coordinate as a distance from this point to a point representing our sample, we'll go from 2D to 3D with a linearly separable classes. And there are multiple standard kernels for support vector machine that increase dimension and increase separability. In some cases, we can go even to a space with infinite number of dimensions. So linear version of support vector machine is just the simplest one. In all cases, this type of classification performs linear separation, but sometimes this is linear separation in another space, in a space with another number of dimensions, which leads to nonlinear separation in the initial space. Another way of classification is related to so-called decision trees. In this case, we have just a tree of decision. At the root level, we look at the first question, the question associated with this root level with a binary answer. For example, for transcriptomic gene signatures, we can see if an expression level of a gene is high enough and low enough, and then depending on an answer, we go to the next node of the tree. At the next node, similarly, we can have a next answer, and step by step we go further and further, and the leaves of the decision trees are classes. So after going through all the tree, we have an answer of the classification. We have a class, well, we have a predicted class for a sample. Due to the fact that overtraining is observed in many cases when decision trees are constructed, the next approach here is to construct random forest, that is, to construct many decision trees and then use voting to select the final class to make the final prediction. Additionally, constructing a random forest, constructing a set of decision trees also provides, well, an approach for estimating feature importance, as it will turn out that some features are present in many trees, some features are almost absent, some features are present in a few trees, so they're present in these trees at different levels. So having random forest, or constructing random forest, we can additionally try to estimate, well, the importance of individual features for the resulting classifier and for the classification itself. Clearly, there are many other ways of constructing a classifier, so a great variety of supervised machine learning methods can be used here. But in all the cases, gene signatures do not utilize the whole totality of genes, the whole totality of features that can be potentially used. They utilized only a relatively small subset of genes. Well, for example, oncotype DX for breast cancer utilizes 16 main genes plus 5 reference genes for PCR. Well, MountPrint uses 70 genes, oncotype DX for colon cancer uses 7 genes. The reason for this is clear. First, but not the most important, it's more simple to measure lower number of, well, to analyze lower number of features and not to perform genome-wide screening each time. But the main reason is the following. If we're using supervised machine learning methods, we need a sufficiently large training set. We need a sufficiently large learning set, that's also called training set. Sufficiently large means that standardly there should be, well, more, in many cases, one order of magnitude more samples than the number of coefficients, the number of internal parameters we should estimate while training. And the standard size of a training set in, well, in the majority of experiments of medical researchers is just several hundreds, well, up to a few thousand. So we can use this training set to deal efficiently with the sets of genes comprising of tens of thousands of genes. And standardly, we're aimed at selecting just, well, for example, 10 genes or even less to perform efficient classification, as for this number of genes, the size of a training set available in the majority of experiments would be sufficient. So it turns out that feature selection is even more important than a method for constructing a classifier. Yet, there are no, well, so standard ways for efficient feature selection. There are standard approaches, for example, a biological approach to select genes that are biologically relevant to a question of interest. So in fact, it just delegated a mathematical task and mathematical problems to biomedical experts. So it really simplifies everything for bioinformaticians, for mathematicians, but it complicates the things for biomedical experts. A simple mathematical approach is to simply select top n genes. If we have two classes, we can perform standard differential expression analysis, for example, and to select the first 10, the first 20, 50 genes as genes that will be used in a gene signature that is constructed. If there are more than two classes, one can use one versus all approach. It means that we perform differential expression analysis, the first class versus all other classes, the second class versus all other classes, and so on. And in each comparison, in each analysis, we select top and differentially expressed genes. Alternatively, we can perform all pairwise comparisons, we can perform differential expression analysis for all pairs of classes and select top n genes from these comparisons. For example, top n approach has been very successfully utilized in constructing PAM50 gene signature, efficiently used for breast cancer patients. Yet there are multiple drawbacks in this scheme. One of the main ones is the following: genes are not independent, and there are many genes which expression levels highly correlate. And if we select one of these genes, if one of them belongs to our gene signature, then in many cases a gene with a highly correlated expression level will also have very high differential expression level and will be also included in top n genes. So we do not select independent predictors. In this case, we do not add much information with new genes, as in fact they contain the same information as information already stored in the genes we've selected at higher positions of a top n list. So there are approaches that penalize correlations and try to avoid this drawback, that go through this list and analyze and take into consideration not only differential expression characteristics, but also characteristics related to linear dependence, or for example, related to correlations with genes that have already been selected. So another approach that tries to address the problem of absence of independence, that try to address the problem of high correlations, is a greedy approach in which we simply construct gene signatures, well, initial gene signatures. We simply make an exhaustive analysis of all individual genes and take a gene with the highest, well, with the highest result, for example, with the highest level of differential expression. But then we take this gene and we again perform the exhaustive analysis. We try to consider all gene pairs in which the first gene is already known and the second one is selected. For each gene pair, we can estimate the cumulative informative power of this gene pair. For example, we can construct a classifier based on this pair and estimate its precision characteristics, and then we select a pair with the highest cumulative informative power. Then, in a similar way, we can proceed from a pair to a triple, from a triple to a quadruple, etc. But there can be additional steps related to removing genes. For example, if we construct a triple, then probably the first gene or the second gene that was optimal at previous steps is not optimal when combined with the third gene that is selected. So in the strategy that uses removing genes in addition to adding genes, when we have, for example, a triple of genes, we can perform the following exhaustive analysis. We fix the second and the third genes that have been selected. So the simplest approach to select genes for gene signature, simplest for mathematicians, for bioinformaticians, is to delegate this task to biomedical experts. So to ask them to give a list of top biologically relevant genes. So it's a real simplification for bioinformaticians, for mathematicians, but it's a real complication for biomedical experts. The next approach is purely mathematical. It's the simplest mathematical approach, called top-N approach. We perform standard differential expression analysis in case of two classes and simply select the top-N differentially expressed genes. If we have more than two classes, then there are multiple ways of implementing top-N approach possible. We can use one versus all approach when we perform differential expression analysis for the first class versus all other classes, for the second class versus all other classes, and so on, and to select top-N genes from the list acquired in each of these comparisons. Or alternatively, we can perform all pairwise comparisons, the first class versus the second, the first class versus the third, and so on, and to select top-N differentially expressed genes in each of these comparisons, and then to combine these top-N lists into one that will be used by gene signature. Top-N approach has been very successfully utilized in many cases, for example, in constructing a very well-known gene signature PAM50, also focused on breast cancer patients. Yet, this scheme has drawbacks due to the fact that genes are not independent. For example, expression levels of genes are not independent. And if there are two genes with highly correlated expression levels and one of these genes is differentially expressed, is really differentially expressed, then, due to the high correlation, the second gene generally would be also differentially expressed, would also have high level of differential expression, and then would also be selected in the final top-N list. So each of these genes, each of these features is informative, yet they are not independent, and adding one gene to another does not give additional information. So in fact, they give us just the same information. And it makes more sense to add a feature with probably low level of individual information in it, but with high amount of new information brought to the gene signature by this gene. So there are some strategies that penalize correlations or penalize linear dependence during the selection of genes. So methods analyzing differential expression tables, which take into consideration not only differential expression parameters, fold change or p-value, but also take into consideration parameters associated with linear dependence associated with correlations of genes. One method to address correlations is to use a very simple greedy strategy. That is to make an exhaustive analysis of individual genes and to select one best gene, for example, to select a gene with the highest level of differential expression. Then to fix this gene and to perform the next exhaustive analysis to analyze all pairs of genes with the first gene selected and to select the best pair. Well, it's a question how to define best. For example, we can construct gene signatures based on each pair, well, compute precision characteristics of for all these pair-based gene signatures and to select the best one. Then to fix these two genes comprising the selected pair and to perform the exhaustive analysis to analyze all triples of genes with two first genes known, and in this manner to select the third gene, the fourth gene, the fifth gene, and so on. A complication of this strategy that's important in certain cases is to additionally remove genes, not only to add, but to remove genes from already selected combinations. For example, if we have a triple of gene, then changing the first gene may be beneficial. It was the first gene selected without knowing the second and the third one, so it's the third gene that's optimal when considered independently or when considered as one gene, but it can be inoptimal in combination with the second and the third one. So if we have a triple, we can fix the second and the third gene of this triple and performing an exhaustive analysis to add a gene for the first position that gives an optimal triple with this first and with this second and third gene, then to try to change the gene on the second position, and so on. Well, clearly it would be much better to avoid greedy strategy and simply to perform an exhaustive analysis to analyze all, well, pairs, triples, quadruples, or whatever size of a gene signature is considered to be optimal in an analyzed question. But in fact, even using supercomputing power, it's feasible to analyze all pairs. It's less feasible to analyze triples, yet in certain cases it seems to be possible. But if we are performing genome-wide analysis, even the exhaustive analysis of quadruples is infeasible, and it won't be feasible in the nearest, well, years, decades, and so on. Well, but as I told, for pairs and even in some cases for triples, exhaustive analysis can be applied. And the application of this exhaustive analysis reveals an interesting fact that indeed exhaustive analysis of pairs proved that indeed informative power or quality of gene signature does not depend only on individual informative power of genes comprising a gene signature. So informative power of a gene signature is not a simple sum of informative powers of genes comprising this signature. It means that there are many high-quality gene signatures, in some cases optimal gene signatures, that would be definitely missed if top-N approach to the construction or a greedy approach to the construction is used. It can be illustrated by the following artificial example. We have two genes the first gene has a relatively high level of differential expression, yet known expression level of this gene does not allow us to perform a perfect classification. Medium expression levels are observed in the black class as well as in the white class. The second gene has very low level of differential expression, so its expression is almost the same in the black class and in the white class. Yet, if we combine these genes, if we consider each sample as a point in a two-dimensional space generated by expression levels of the first and the second genes, we'll get a perfect linear separability of classes, so perfect separation, perfect classification will become possible. In standard top-N approach, we won't select gene 2, we won't include it into a gene signature as its differential expression characteristics are very poor. Yet, in combination with the first gene, gene 2 becomes very efficient. As I told, it is just an artificial example, but there are real data examples, for example, in breast cancer related data that also illustrate this fact. So, for example, in this case, expression level of gene IGF-BP6 is almost the same in the black and in the white classes. Well, E-level5 gene has slight differential expression, but it's really not so impressive, and E-level5 would not be selected as the first gene in a greedy strategy. It won't be selected in the top-N approach. So it does not belong to the top, I think, 100 or even more genes if the genes are ranked based on differential expression characteristics. Yet a combination of these genes still allow very good separation of patients with and without recurrence in the first 5 years. So just to summarize what we're talking about today, gene signatures standardly comprise of several genes and are used for diagnostics, prognosis, and prediction. So the main problems, the main types of gene signatures are diagnostic, prognostic, and predictive. Gene signatures generally have precision characteristics that are far from 100%. In many cases, that they have characteristics that are far from 100%. Well, mainly these are prognostic and predictive gene signatures that have relatively poor characteristics. Yet gene signatures are very important as an additional source of information for making a decision. So a key question in constructing of a gene signature is selecting features. And when we have features selected, we can utilize multiple machine learning approaches to finalize a construction of a classifier. In many cases, selecting features and constructing classifiers go in parallel. For example, in a greedy approach, when we estimate the quality of a gene set, of a gene pair, gene triple, gene quadruple, so on, by constructing a classifier and analyzing its precision characteristics on subsets of a training set. So that's it.